Hi, everybody. We're jumping into Chapter 7, Neoplasms. The textbook does a really nice job, I think, with giving us an overview of cancer pathophysiology. And then we are going to tie in from our Osmosis Elsevier videos our specific cancers that we're going to kind of hone in on a little bit that way. So we're going to concentrate on two parts. First part's going to be the cancer pathophysiology, and then the second part will review a couple of different cancers that we want to kind of highlight. As always, Tax goes straight into the deep stuff of the pathome. Key concepts. Cancer is essentially unregulated cell proliferation. So our cells are turning over and creating these tumors that are not normal and they're bad. They usually originate from one or a small number of DNA mutations due to environmental exposures or just purely by random chance. Unregulated cell proliferation reduces the DNA protections, and that allows the mutations to continue to grow and accumulate. In many cases, mutations facilitate invasion across basement membranes and metastasize to different sites. We're going to talk about proto-oncogenes, which are codes for proteins that are needed for rapid growth and development. They're generally turned off after childhood, but if you have a genetic mutation that turns them back on, they can become oncogenes. The oncogenes are code proteins that promote repeated cycles of cell division that escape normal growth limitations. The tumor suppression gene is a code for proteins that stop the cell cycle, preventing cell division when a cell has abnormal DNA or some other pathology. So these are kind of our checkpoints of how we're supposed to be able to prevent these things from happening. You also can have an inherited predisposition to cancer. However, it's fairly rare, and actually only less than 5% of cancers are related to genetic inheritance. Inherited mutations that promote cancer involve tumor suppressor genes, which are the proteins that stop the cell cycle, preventing division when a cell has abnormal DNA. So most of these will involve some manipulation of tumor suppression genes. When one allele is mutated after birth, cancer does not develop. But a second hit then is required, generating a mutation in that second allele, then at which point that protection becomes lost and cancer can grow and spread. Gene mutations accumulate in cancer cells due to our genetic instability. Tumor cell genes are tracked during the course of treatment and used to guide chemotherapy choices. So what I'd like for you guys to do is to jump over to Osmosis, and I will add the link here. So I've added the link there, and that cell cycle is on your Chapter 7 playlist. It was on some of the previous chapters as well, but I do think if I were you, I would stop and review that and listen to it again. Then come back and jump on to the next slide. So hopefully you've watched that cancer or the cell cycle Osmosis and you're back now listening. A quick overview of cancer pathology and pathophysiology in America. We did already talk about. Sorry, I have a frog in my throat. In the United States, cancer remains a leading cause of death, with nearly 1 in 4 deaths resulting from that disease. So this is obviously something we are going to see come across our doors in practice. All right, so we jump right into then the cell cycle. So that is a complex system. These are all these different interacting molecules. We are multicellular organisms, so we need a coordinated cell division, right? We need a coordinated, controlled process to continue to maintain cell growth, and we also need controlled cell death. So the rates of cell division will vary widely among all the different species and all the different cells in our human body. The example is mature cardiac cells and neurons exhibit low rates of division, whereas cells lining the GI tract and the blood cell, precursors of bone marrow will divide rapidly. So we use the words like rapid proliferation when we're talking about cancer quite often. So the steps of the cell cycle must proceed in a very careful and regulated manner because we have to have proper production of these new cells. And when things go askew, that's when we get cancer. Consequently, the cell cycle exhibits distinct phases, each with its own molecular signature and specific function to accomplish the generation of viable new cells. So, to achieve this, our body not only manages our own internal machinery within the cells, but we integrate cues from our extracellular environment. So we need the influence of growth factors, we need availability of our nutrients in our body, right? And we need to have physical interactions with neighboring cells. During this process, this tight regulation of our cellular proliferation must exist to avoid pathology. Off the rails, then we're going to have a disease. All right, in its simplest way, we can divide the cell cycle into two broad categories. We can have interphase, which describes the period in which the cells grow and replicates its DNA, and that is necessary for cell division. And then we can have M phase, which is the mitotic phase, which entails the separation of chromosomes and cytoplasm. So for cells to prepare for division in the interphase, they have to carry out this division in the M phase. So this image here is in your book as well, that is showing the two major phases. I jumped over, sorry. That slide, thank you. This one here is showing the two major phases of the cell cycle. This one starts with the interphase. So A here is interphase. So during this, the cell prepares for division. In G1, the cell grows, the organelles begin to duplicate, and RNA and protein synthesis are increasing to complete the replication of DNA in the S phase. The integrity of DNA is assessed and repaired, if necessary, during G2. So that's what they're showing you here. Right? So we started, the cell grows, the cell starts happening, the DNA is actually replicated, but we have a final step of G2, and that's our checkpoint to make sure we have a good quality thing happening here, right? So in interphase, much time is spent here. We call it the quiescent cells in G0, when the cell is laying kind of dormant. Interphase does include G1, S, and G2. Commitment to divide controls G1 through S. So we have this transition that allows the DNA replication to proceed here. G2 permits the assessment of the DNA. So this is that strong checkpoint. It's the guy with the clipboard at the end of the factory before the thing leaves the factory. And it's making sure that the cell contents to build two daughter cells before mitosis can begin. Okay? And then we go into the mitosis part, which is, seen here in B. The mitotic phase, so we're M phase, right? The details in this phase to process replicated DNA is separated, forming two nuclei. During cytokinesis, the contents of the cell are separated as two new cell forms. So now we've finished G2 when we go into M, which is here, and this is cytokinesis, and the cellular contents are now officially divided. So this actually shows us the cell mitosis and the cytokinesis and what that looks like here. This process in M phase of mitosis, right, involves the condensation of DNA into chromosomes during prophase, while centrosomes move towards the opposite end of the nucleus. And it's showing us this here. And during cytokinesis, an actin and myosin ring aligned at the center of the dividing cells begin to contract, and that actually separates the cellular components and seals off the membrane of each new cell. Okay. So in your book, mitosis is seen in the 5 distinct subphases, which is prophase, prometaphase, metaphase, anaphase, and telophase, and they are described in box 7.1. So if you go to, I'll add that as a slide here, box 7.1, mitosis subphases is worth spending a little time on, right? Oh, I thought I had slides for that. Just ignore me. There we go. Okay. Prophase. So during the initial step of mitosis, prophase, the chromatin condenses into chromosomes. This is a process that involves regulated compaction of DNA. But remember, during prophase, this is where the nuclear membrane remains intact. A protein complex assembles on each sister chromatid at specialized regions known as the centromeres. And these structures will be important because remember, those sheaths will pull apart, and that's what actually causes the separation. Okay, but at this point, they are assembling on the sister chromatid. In each cell, the microtubules are now organized in a structure known as the centrosome. And the centrosomes duplicate during cell division and move towards the opposite sides, right? Because they know they're about to be split apart. But in prophase, the nuclear membrane remains intact. Then we go into prometaphase, and the nuclear membrane breaks down and exposes the sister chromatids to the microtubules of the centrosome. Okay. Microtubules from the opposite centrosomes will bind to the protein complexes of each sister chromatid. They form this very tight connection, and now we're creating tension. So in my mind, prophase, the nuclear membrane is remaining intact, and those tendrils are beginning to move towards the outside of the cell. But in prometaphase, we actually bind and form tight connection with tension. Then we go into metaphase, and now we are working with moving the sister chromatids towards the center of the cell, and that can create an appearance referred to as a metaphase plate due to the alignment of the sister chromatids along the center. Okay. What I think of for metaphase is that this is where now everything has to realign properly before we can move to anaphase. And in anaphase, okay, everything is now aligned, and the proteins linking the chromatids are released, and those bound microtubules begin to shorten during anaphase. And as that occurs, the chromatids are effectively separated and pulled to opposite ends of the cell by the shortening microtubules. That allows us then to go into telophase. So now you have this separation, and now the attached microtubules disassemble, while two nuclear membranes now need to form around the now separated chromosomes. So at that stage, the replicated DNA has been split, and it's moved to opposite ends. Okay? But the cell must still effectively divide the cytoplasm. One important part to remember is that while the replication is happening, the replicating cells will move through additional rounds of the cell cycle, but the cell may leave progression and enter into G0, right? And in G0, the cells remain viable and functionable, but they're not actively doing anything. They're quiescent and they're in rest. So a cell may permanently leave the cycle and enter G0 once it's matured and has performed its specific role. So this is an example of terminally differentiated cells. They've done their job, they're done. Most cells have a finite number of divisions that they can complete at the end of their chromosome, because at the end of the chromosomes, those telomeres will become progressively shorter with each division, and that is replicative senescence, okay? Well, that makes sense. So what we're saying is that they can only replicate so many times before they have to go senescent. They have to go quiet. They have been terminally differentiated. They're kind of done and they've served that. So once that critical stage has been reached, the DNA can no longer be appropriately replicated. And so they have to enter into a permanent state of G0. So the book talks about them being mainly like examples of heart and brain cells. And then they give us the example of liver cells, which have a much higher capacity for replication and growth, because we can regrow our liver after injury, right? We don't regrow parts of our heart after injury. The liver cells have the capacity to exit out of G0 and re-enter the cell cycle. So cells possessing that ability are said to be quiescent in that they can enter G0, but they can later resume the cell cycle if necessary and return to G1. The other example of this is memory T cells of our immune system. So during exposure, when they're needed, the pathogen's been seen, the T cell needs to be activated, and it needs to increase in number as part of our immune response. So it's going to exit G0 and go into cell phase G1 to begin to replicate itself because it's needed. Following the resolution of the infection, the portion of these cells remain in the body because they go to state G0. And if we are exposed to that pathogen again, they're going to come out of G0 and continue the cell cycle. So some cells have replicative senescence, meaning there's only so much that they can do, and then they're going to be kind of terminated, so to speak. And other cells can come in and out of G0 back into G1. So how do we control the cell cycle? What is the factors that are going to prevent cells from entering or exiting both appropriately and inappropriately, right? So this is where we come with the cyclin-dependent kinases and the cyclin-dependent kinase inhibitors. So these are the CDKs. When active, these kinase proteins will phosphorylate a specific set of protein substrates. It's through that phosphorylation that it begins to set in motion the various stages and transition points in the cell cycle. So these things have to be phosphorylated at key times to help move things through the cell cycle. The activity, though, of the CDKs is regulated by their own signatures of phosphorylation. So, as the name implies, it depends on the binding of the proteins, which increase the CDK function. What does all that mean? That they have to do their own phosphorylation to control the different CDKs and their activities. Cyclins, fluctuation in their levels will determine the cell cycle orderly progression, so moving in order, controlled by other proteins at the other critical checkpoints. After completing their role, the cyclins are readily degraded to prevent abnormal cell cycling, right? So again, they have a limited use, and when they're not going to perform well, they are degraded. The CDK, the cyclin-dependent kinases, these are considered the workhorses of the cycle. They're unable to function unless level of their specific cyclin increases. The cyclin-dependent kinase inhibitors can be activated by signals like P2-1 protein and halt CDK activity and cell cycle progression when surveillance indicates that we have a problem. So all of these different proteins are working together. So what we need to understand is with these three critical protein families is that while the cellular levels of CDKs remain relatively constant, the levels of the cyclins will rise and fall as the cell cycle progresses. And that is what we see here. So we have cyclin D levels rise through G1, initiating the increasing cycle E and phosphorylation of P53. P53 is the guardian of the genome, and that prevents progression from G1 if the DNA is damaged. Cyclin E produces the G1 to S transition, and cyclins A and B promote movement through G2 into mitosis. So if someone were to ask you, how does the cell cycle regulate itself to ensure that the steps are followed correctly, we would say, well, through the levels of cyclin that will rise and fall and fluctuate as needed. And we know that the CDKs, though, remain relatively constant. It's also important to remember, though, that we have to have the capability to stop when necessary. So if a cell has experienced an alteration through damage or mutation, the cell must be prevented from proceeding, right? It can't move on to the next step. Cells must ensure that the necessary nutrients and building blocks are present before proceeding through the steps of division. So we have different regulatory things happening. One regulatory protein of the cell division is called the retinoblastoma protein, PRB, and that inhibits the cell from progressing through G1. Okay. As levels of the cyclin D, which we can see here, rise in response to the growth factor, and nutrient-inducing signaling happens, that leads to the change in the structure. The structural shift causes the release of the bound transcription factors and allows the cell to continue. Okay? You don't need to memorize these concepts. We need to have a general understanding of these concepts and what they mean as to what they are doing. So the P53 gene is what we consider to be the guardian of the genome because it's critical to halting the cell cycle when DNA is damaged or the cell has injuries. Okay? So I would think that we would understand that the P53 is obviously something we're going to talk a lot about in neoplasms because there likely is something going to be wrong. Okay? The link between P53 and the suppression of neoplastic growth has been observed and helps support the idea that P53 is part of the cancer process. We've heard of this before. The video talked about it is these checkpoints. They are present throughout the cell cycle to ensure that the division undergoes its right steps and it's done correctly. We have G1 checkpoint, ensures the presence of necessary growth conditions and commits the cell to the remainder of the division process. Okay. Checkpoints are in place to control the progression and to block movement to the next cell cycle phase when DNA is damaged, nutrients are not sufficient, or energy is deficient, or growth factors are lacking. When we don't have these oversight proteins, we're going to have neoplasms proliferate quickly. All right, this is actually, I'm laughing because this still strikes me of we don't use these terms correctly. And if you've ever worked in hematology, oncology, or really anywhere, it becomes really frustrating. And I explain this to patients all the time, and I feel like it's really important that we do that because these are very different terms. So a tumor is any abnormal mass of cells. Okay? An abnormal mass of tissue when the cells divide more than they should or they do not die when they should. A neoplasm refers to new and uncontrolled proliferation of cells that can be benign or malignant. So if you use the word neoplasm, people assume it means cancerous, and it's not. We need to say a malignant neoplasm or a benign neoplasm to be more specific. Not all malignant cellular proliferations are actually going to form a tumor. We can have that proliferation in our hematology parts, right? So leukemias, etc. A benign tumor is typically slower growing, it's well demarcated, and it typically doesn't infiltrate. So when you look at it under the microscope, there's a well-contained area. When you look at it microscopically, the tumor cells do not infiltrate beyond the basement membrane, and they don't go into adjacent tissue. Okay? Dysplasia is a term that describes a change or alteration in the cells. Okay? So they're abnormal appearing, and we worry that they can turn into malignant cells. But a dysplasia in and of itself is not considered a malignancy. The term carcinoma in situ has been used to describe aggregate of malignant cells that have not extended beyond the basement membrane. So typically carcinoma in situ, we think of DCIS, which is ductal carcinoma in situ of the breast. And again, we think that those are actually malignant cells, but they're in situ. They have still been contained. A malignant tumor, however, is composed of cells that are able to infiltrate through the basement membrane and will go into the adjacent structures. Okay, the tumor margins are poorly contained, and microscopic examination reveals infiltration of the tumor cells into the surrounding tissue. That may allow the malignant cells to extend and progress and metastasize either through the lymphatics or the blood vessels, and ultimately they can seed in other parts of the body and other cells, and when they form new tumors, we call that metastasis. Malignant tumors are named based on the tissue from which they develop. This is really important. So a carcinoma, for the example, is a malignant tumor that's derived from cells of epithelial origin. So squamous cell carcinoma, adenocarcinoma, small cell carcinoma. So squamous cells come from the squamous epithelial. Adenocarcinomas come from the glandular epithelial, and then small cells come from neuroendocrine cells. Sarcomas are malignant tumors that arise from the mesentery type tissues, so blood vessels, cartilage, bone, muscle, and they're rare when you compare them to carcinomas. The prefix of a sarcoma designates its origin. So the example they give in the book again is osteosarcoma, which is arising from bone tissue. So the osteo comes before sarcoma, meaning the bone. But like a liposarcoma creates a sarcoma arising from adipose tissue, right? So that makes sense to us. Hematopoietic neoplasms are proliferation of these abnormal cells within bone marrow. It can also be occurring in our lymph nodes, spleen, or liver. When we refer to things like leukemias and lymphomas, we have to understand their origin and where the neoplasm is occurring. So when we talk about a lymphoma, that's going to refer to a malignant, so a cancerous tumor that results from monoclonal proliferation of lymphoid cells. These can be things like B or T cells. So you can have a B cell lymphoma or a T cell lymphoma. A leukemia occurs secondary to the monoclonal proliferation of early hematopoietic cells, which is our blast cells in the bone marrow. And then we get these abnormal early cells that proliferate throughout the blood. The other ones we think about that we may see is a melanoma, and that is from the melanocytes of our skin, typically. We can have tumors of the brain from the multitude of cells in the nervous system. Those tumors can come from the neurons, the glial cells, the choroid plexus, and the meninges. So if we talk about a glioblastoma, we're talking about the brain cells. And the glial ones are very common in the forms of different brain tumors. Remember that you can have brain tumors that have metastasized to the brain from other sites of the body. And then the interesting one to always remember and think about is a teratoma. And that's a tumor that arises from germ cells, and it's composed of tissues derived from one or more of the embryological germ cells. The example that they give is a mature cystic teratoma, which is a dermoid cyst, and that may occur in organs such as the testes or ovaries. Switched my slides around on the side. You can't see. Let me fix that. There we go. Okay, so we talked earlier about the normal cells, the normal cell cycle. Now we're going to talk about specifically some of the things that are important to cancer cells. So as a general principle, the genetic changes that promote this stepwise progression from a normal cell to a malignancy will alter the amount, activity, or regulation of two main types of genes. Proto-oncogenes are genes that possess the ability to promote cellular proliferation and survival, but are subject to careful regulation. Okay? Proto-oncogenes encode proteins that promote cell division. The highest activity is during embryonic development and childhood. And in childhood, this is what helps us to have such accelerated growth. But essentially, they should be shut off when we're not needed. So after we're kind of done with puberty, they should be shut off. But we can have mutations that will reactivate a proto-oncogene, and those then are called oncogenes. So if you think about it, if we have genes that are good in the embryonic development and in our childhood that cause great rapid accelerated growth, that's fantastic, right? Think about how much you grow from the age of a newborn to a 10-year-old. So to have that much change at the cellular level, we really need these proto-oncogenes. But then once we're kind of at our grown, we've reached our kind of stature, we turn off our proto-oncogenes. So if you have a mutation that's going to turn them back on in a cancerous fashion, we don't want those active that are unchecked, right? Because they cause rapid growth. The second significant gene we have to talk about that affects cancer cell creation is the tumor suppression gene. And this one is one that causes loss of function. The mutation in both the alleles cause loss of functional protein, which promotes unregulated growth. And these include cell cycle suppressors and many DNA repair genes. So the term tumor suppressor genes is kind of a larger umbrella. Many protective genes encode proteins that inhibit uncontrolled cell division and do the surveillance of DNA. So any of those protective genes could be referred to as tumor suppressor genes. I cannot speak today. This is figure 7.5 in your book, and this is showing, again, that we have these two major groups of genetic mutations that will help progress a cell to malignancy. So A, we have a proto-oncogene within a normal cell is mutated and it turns into an oncogene. So an oncogene has been created. The oncogene produces excessive and unregulated cell division. So then we get to B, a tumor suppressor gene is inactivated by a mutation. The remaining normal allele sustains cell protection from proliferation, but if the second allele develops an inactivation mutation, the tumor suppressor function and its pathway of protection from malignancy are lost. So this shows how a cell can become a cancer cell. Normal cell has a single mutation event, so the proto-oncogene created an oncogene. Activating mutation enables oncogene to provide or promote cell transformation. Also, we have a normal cell that has a mutation event that inactivates a tumor suppressor gene. And it doesn't affect one copy here, but it affects the other copy. And if a second mutation event occurred, it will inactivate the second gene as well. So now you have two inactivated mutants functionally eliminate the tumor suppressor gene promoting cell transformation. So these are the two paths that we can alter cells most typically to become a cancer cell. Cancer cell characteristics are hallmarks of cancer cells, okay? They have uncontrolled proliferative signaling. So remember, that can be initiated by the many different types of oncogenes. They can evade growth suppressors, and we can have genomic instability. Cancer cells can have replicative immortality. I love that word. The telomerase grows ends of chromosomes, avoiding that cell senescence. Remember when we said that the cells are protected and they're only able to replicate so many times? before they become senescent and they turn themselves off and they go over to G0 and there they stay. The cancer cells will continue to grow the end of the chromosome and they avoid that altogether. This is supported by the fact that many cancer cells lines derived from cancer patients can survive indefinitely in the lab if they are given appropriate growth conditions. So, if the telomerase will continue to cause us problems, and currently in cancer studies we're trying to find telomerase inhibitors to try to be an anti-cancer drug or drug treatment. Cancer cells can resist cell death. And this occurs through two primary routes. Okay? Necrosis represents the uncontrolled form of cell death. It often occurs in response to acute cellular injury. The plasma membrane ruptures and the intracellular contents will spill out into the surrounding tissues, and we get the inflammation and we get tissue damage. Apoptosis is regulated and programmed form of cell death. I think of apoptosis like our current iPhones. They're only meant to last so long and then they're just going to die. Under normal conditions, cells that sustain extensive or unrepairable damage to their DNA, they will undergo apoptosis due to our intracellular surveillance mechanisms. So they undergo scheduled death. Abnormal or pathogen-infected cells will also be targeted for apoptosis or outright lysis by our immune system. So, apoptotic mechanisms serve as a protective function in the body. However, cancer cells have found ways to avoid and evade apoptosis by upregulating anti-apoptotic factors such as IAPs, inhibitors of apoptosis, or they decrease the production of pro-apoptotic factors such as FAS. What we're saying is that this process enables cancer cells to resist apoptosis, avoiding this built-in mechanism of cell death, right? They bypass that, allows a cell with mutated DNA to continue to grow and divide, and they make themselves more tumorigenic and ultimately malignant. The next hallmark of a cancer cell is angiogenesis promotion. So as the tumor grows, they require blood supply and production of the vascular endothelial tissue. The production of vascular endothelial growth factor provides the tissue new capillaries to nourish the tumor. So that's a pretty darn smart cancer cell to me. They're like, ooh, if I create these new blood vessels, I can even hyperfuel my cancer cells to become even more active, right? So this is really, really tough. So in cancer realm, you may hear of VEGF. That's the vascular endothelial growth factor. And molecules such as these really, really are hard to do. But we now have drugs. Some of our monoclonal antibodies, for example, are VEGF inhibitors. Bevacizumab, which is Avastin, is a drug that's really commonly used, but it's a VEGF inhibitor. So it is used in the treatment of cancers that have metastasized or recurred. And what essentially that drug is going to do is turn off angiogenesis of the tumor. It kills its blood supply to help kill off the tumors. That's pretty cool. Okay, next. Cancers are unique because they have invasive and metastatic capacity. So, cells revert in an epithelial mesenchymal transition. Adhesion to extracellular matrix loosens, and the matrix-degrading enzymes are expressed, and the cells acquire motility, which is a problem, right? I think of angiogenesis as like pouring extra fuel into the fuel tank. And now this is the EMTs actually put tires on the cancer cell. So in order to metastasize, the cells have to separate from the original tumor. That makes sense. And then they have to invade the tissues. They have to enter and survive in circulation or the lymphatics or our peritoneal space. And then ultimately they have to settle in a distant target organ and then they have to adapt, survive, and proliferate. Okay, so they're busy little bees. To do that, metastatic cancer cells typically will develop alterations in their shape and in how they attach to other cells and how they form their extracellular matrix. And this is where we get this EMT. And it's an important process because it allows transformed epithelial cells to invade tissues, to resist apoptosis, and to ultimately spread. The example your book gives is E-cadherin, which is a key adhesion molecule, and it's lost in many cancer cells. This allows the tumor cells to detach from the surrounding cells and become free and can float around and help support invasion and metastatic processes. We have new classes of anti-neoplastic drugs, such as chimeric antigen receptor T cells. So this is CAR-T. This is something I see quite a bit in my cancer patients, that they are on CAR-T treatment is how they'll be referred to in the notes. And these are drugs that are engineered to attack an individual patient's tumor, checkpoint inhibitors that permit the lymphocytes to proliferate on stimulation. These hallmarks are now considered part of the core hallmarks, and more recently emerged hallmarks include the microbiome, epigenetic reprogramming, and phenotypic plasticity. I also wanted to point out in here that in addition to the CAR-T being an immune-based treatment, we also have the monoclonal antibodies, and several types are specific to use in cancer. Monoclonal antibodies can opsonize tumor cells, marking them for phagocytic destruction. At the same time, destruction of the cells increases the reaction of antigen-presenting cells, or APCs, that can drive T helper and cytotoxic T lymphocytes mediated tumor destruction. What does that mean? So not only when you give a monoclonal antibody, not only is it actually going to opsonize the tumor and let the body know it's ready for phagocytic destruction, but the destruction of the cells then also increases the reaction of the APCs. And so we have more ability for those APCs to help deliver those antigens to the T cells, right? And we can help facilitate tumor destruction even more. And I bring this up because these are really highly effective drugs, and they have really changed the landscape of cancer treatment. Because we now have immune-based cancer therapies, it has completely altered the playing field in a really good way. We now have ability to have people live longer and people with metastatic disease to live longer. So these are really big breakthroughs in cancer technology. Next, they move on to talking about the metabolic alterations of the cancer cells. Remember, a growing tumor will have varying degrees of oxygen supply. It depends on how close they are to the previous blood or to existing blood vessels, or if they have a type of tumor that's already forming new blood vessels. So depending on where they're located, they'll have varying degrees of oxygen. The collection of cells will consist of tumor cells in various states of genetic alterations. Each will have somewhat of a different characteristic, a different energy requirement. Plus, the surrounding tissue cells, called the stroma, will have varied impact on the alterations. So cell types making up the tumor will include the cancer stem cells that propagate their own growth. We will have those immune cells that are stimulated by the inflammatory signals of the tumor, right? So when we're talking about kind of the microenvironment around them, what's all involved in this area when you look at it, we have the cancer stem cells, the immune cells that are coming now because of they've been stimulated by inflammation, so lymphocytes, macrophages, dendritic cells. Then we also have fibroblasts and cells undergoing the EMT. These are the cells that are trying to get their wheels, so to speak. The microenvironment of the tumor then depends on the interactions between all these cell types, and that will help determine the type of metabolism that's needed by the cells in different stages as this tumor progresses. Okay? So one theory or model proposes that hypoxia-stressed cells in the center of a tumor adopt the properties of increased survival, treatment resistance, and immune evasion. And ultimately, they will become more likely to develop mutations that favor invasion and metastasis. So that is seen here. A tumor is then made up of their stem cells, their cancer cells, the original tissue or the stroma that was there in the first place. Now remember, we've had inflammation, so we have lymphocytes, macrophages, and dendritic cells have all rushed to the area, and cells in some phase of the EMT. Some of those are beginning to learn to grow with their wheels and become mobile. The tumor region closest to blood vessels will typically have a normal oxygen supply, right? The cells that are near the normal oxygen supply have cells that maintain normal sensitivity to killing by our CTLs. The tumor region at a distance from the blood vessel has a hypoxic and acidotic environment that induces cell stress, right in here. This may promote acquisition of cancer cell immune resistance and development of immune tolerance, and can help favor mutations that ultimately enable invasion and metastasis. Okay. And in here, they tell, some of these tumor cell adaptations, right, are listed here. And then you have this breaks this barrier, and we can get the invasion and metastatic cells. So the point of all this is that stressed cells become more aggressive and are more likely to be invasive, which is not a good thing that we want, right? So we will have local and systemic manifestations of cancer, right? So when we say what is the clinical presentation of patients with cancer, what are we going to be looking for? Or what kind of signs and symptoms are your patients going to have? What are they going to be complaining of that would trigger us to think, I worry about a cancer? So we can have both local and systemic consequences of cancer that are going to cause signs or symptoms, okay? And one of those is obviously inflammation. So with inflammation, we can get fevers, but we can also suppress appetite, and we can also promote clotting. So some patients will present to the hospital with a DVT or a pulmonary emboli, and that was how we knew they had a cancer. Or some people have just been losing weight. They just don't have an appetite, and even though they're eating some, they're just losing weight. Or some cancers present with low-grade fevers or things like night sweats, fevers at night, right? Hypercoagulability can accompany cancer, particularly in early stages. So that's tough. Also, fatigue is very common, particularly in advanced cancers. Poor appetite and wasting, so not just weight loss, but also loss of muscle can occur in advanced cancers as well. Endocrine-related syndromes can result from tumors that are ectopic sources of hormones and hormone-like substances. So we can get things like SIADH, right? So we've got some of the things we need to think about could be systemic manifestations of cancer. So if you came across somebody with abnormal sodiums and you diagnosed them with syndrome of inappropriate antidiuretic hormone, or SIADH, one of the differentials we should have in the back of our mind then is the possibility of a cancer. Okay, so talking now about some of the other things you may see in clinical practice. One of them is tumor lysis syndrome. We have tumor lysis syndrome that can happen when we don't know what's going on, and then we can induce tumor lysis syndrome because of the treatments that we give. Tumor lysis syndrome is an acute generalized reaction to massive cell death caused by a cancer treatment. Although it can be spontaneous, it generally, most of the time, is precipitated after initiating a round of treatment that is robust and kills a lot of cells very quickly, which sounds awesome, right? Don't we all want to have a treatment that's super powerful and has this massive killing of cancer cells? But the cells then also release their contents, and the contents of those cancer cells are problematic. And you can have too much uric acid, too much potassium, too much phosphate, and not enough calcium as a result. So you can get these electrolyte imbalances. And those then can cause instability of excitable tissues. You can get cardiac arrhythmias, and you can get seizures. Also, having to ask the kidneys to handle that much quote-unquote debris can cause acute renal failure. So we kind of guesstimate the risk for that by looking at the estimated tumor volume, the cell lysis potential of the treatment, and patient factors such as their fluid and electrolyte and their renal status. So oncologists and hematologists know which kind of treatments typically can cause tumor lysis syndrome, and we kind of risk stratify for it. And so oftentimes they are in the hospital and they're getting lots of fluids and lots of allopurinol and frequent lab monitoring. The other things that we can see that can be cancer or therapy-associated complications: pain, anemia, abnormal blood counts, nausea, vomiting, stomatitis, fatigue, radiation-induced tissue injury, they cannot sleep, and dysphoria. So when we're thinking about new cancer agents and anti-cancer agents and cancer treatments in the future, we really want to make sure that people are also focusing on drugs that can be highly effective, but also would have limitations of some of the adverse side effects, or what we're working towards. And that's what I mean by immune therapy. They're a lot more readily tolerated than our previous cytotoxic chemotherapy. When we get to pathophysiology, we'll talk more about the cell cycle again and some of our chemotherapy agents. The biology of cancer risk factors and mutations. Remember, we said at the beginning that we do have some genetics and inheritance in cancers, right? So the cancers can result because of an inherited gene, but they also are caused by bad luck, essentially. When it's a germline mutation, those are mutations that are present at birth. They are actually a minority of cancers. Inheriting a germline mutation will result in every cell of the body will have that mutation. Somatic mutations are mutations that occur in the cells spontaneously or from exposures to something. So you can have germline mutations, which literally mean germline of like eggs and sperm, right? So germlines we have at birth, and they're in all our cells. Somatic mutations occur as a result to an exposure to something. And a somatic mutation is inherited by the progeny of the cell with the mutation, but it doesn't occur in all cells of the body, and you can't give it to an offspring. So it is much more common to have sporadic gene mutation or somatic mutations rather than inherited mutations. Really, it's about 5% of cancers are inherited mutations. Also, though, uniquely, sporadic gene mutations can accumulate in the tissues over time and then develop a tumor. The example the book gives is a very common thing called benign adenomatous polyps of the colon. And the colon accumulate gene mutations over time, and then those can develop into a malignant tumor. So that's why when they take some of your polyps from your colon, you may get a report that says you're at risk of developing cancer with these things, because if you get them enough and you aggregate enough in that area, they could turn malignant. So that's why we do colonoscopies, right? We have to know that ahead of time. But we have to remember that other risk factors are at play to help someone develop cancer, right? And like one of the first things we think of is smoking, right? If you're perpetually exposing the lung tissue to that carcinogen, it is highly risky that you're going to get a cancer. The other one is alcohol consumption. Sometimes we don't really understand it, but race and age and ethnicity can be at play. Obviously, exposure to UV light, and then other environmental toxins. We also know, though, that just lack of general health and lack of exercise and being obese are also risk factors for cancer. Which are there. Okay, so some of the hereditary cancers we do need to talk about that you've heard of them again and know what they are and you're familiar. One of them is Lynch syndrome, hereditary non-polyposis colorectal cancer. It's a defective mismatch repair gene. Okay. The mismatch repair genes are genes that will play a role in ensuring the accurate pairing of our DNA base pairs. And so when those go awry, we can get Lynch syndrome. Microsatellites are these short DNA sequences that may be altered due to mutations in the mismatch repair genes, and that may result in genomic instability called microsatellite instability. And you may hear of this especially in the colon cancer world. Okay, the next one is Isai-Fermini syndrome, or Li-Fermini syndrome. And again, this is that P53 gene mutation, right? So in this syndrome, it's probably Li-Fermini. The germline mutation is a tumor suppressor gene, T, tumor suppression of P53, is inherited. So patients with this syndrome are at an increased risk for the development of tumors at very early ages. Those are things like sarcoma of the bone or soft tissue, even breast cancers, brain tumors, or tumors of the adrenal cortex. And then, of course, we know of BRCA1 and BRCA2, and these are product functions like tumor suppressor or DNA repair proteins. And we know that if you are a BRCA1 or a BRCA2 carrier, you're at risk for an increased risk of breast, ovarian, fallopian, or prostate cancer. So yes, men can be checked for BRCA1 and BRCA2. But remember, just because you carry the gene mutation does not guarantee you're going to have it. You still have to have the second hit. So it's like if you had the two alleles next to each other for your DNA, and one was already kind of broken, it's not going to take much to damage the whole set if the other one even gets a little hit. So now both parts have taken a hit, and now you're going to get the cancer. So just because you have a BRCA1 gene does not mean you're guaranteed to get the cancer. However, in your lifetime, you're going to be at a much higher risk because it's only going to take one certain type of exposure, and you will have cancer. Oh, I did also want to point out on here of the Philadelphia chromosome, right? So this was the first kind of identified chromosomal translocation, right? And we call it the Philadelphia chromosome. The translocation is visible upon karyotyping and labeling, and this is BCR-ABL fusion gene and whatever. But if you ever hear about the Philadelphia chromosome, it's figure 7.7 in your book, and it's kind of cool because that's something you may hear along the way. And it's there on that slide. The CML can be caused by Philadelphia chromosome, and then the abnormal BCR-ABL can also cause some leukemias. All right, moving on to viruses. Viruses can cause cancer. What a bummer, huh? We're learning more and more about this. This is like why the HPV vaccine has come out, right? Because we realize that exposure to these viruses are associated with increased risk of cancers. The ones they talk about here is the Epstein-Barr virus. If you've had that before, it's Burkitt's lymphoma and a B-cell lymphoma. Hepatitis B can be associated with hepatocellular carcinoma. And then HPV. HPV is a small sexually transmitted DNA virus. and there are more than 200 genotypes of HPV. However, when we give you vaccinations against HPV, we focus on ones we think are most likely to lead to cancer. So subtypes 6 and 11 are associated with lesions in adults and lesions in children, but the subtypes 16 and 18 have been implicated in the development of head and neck and anogenital cancers. So that's why we created the vaccine for HPV. If we can prevent you from even getting HPV, we can decrease your risk of getting those kinds of cancers. And what they're saying here is that hepatitis B and HPV are preventable by vaccines. Launching into genotyping and cancer diagnosis and treatment. This is a huge area of study, and it has come such a long way in the last 30 years. So the approaches to the treatment of cancer is obviously going to depend on the type of cancer, where it's located, and how far it's progressed. The three main typical things we think of for treatment of cancer is surgery, radiation, or chemotherapy. Remember this, surgery is only helpful in a solid tumor, and it's most helpful if it has not progressed or metastasized. So if you have lung cancer, but it's already spread to your bone, us going in there and cutting out the lung cancer is not going to be helpful at all, right? But if you have a small tumor in your lung and it hasn't progressed anywhere, now it makes more sense to go in and do a lobectomy. Given that cancer cells often have fast growth rates, chemotherapy can be used as a non-specific target to any rapidly growing cells. Well, that's why we can get a lot of systemic complications from chemo as well. Chemo is usually reserved for advanced cancers that have spread from their primary site of origin. Okay. So we are trying to refine our treatments of chemotherapy, and we're trying to use more things like immunotherapies, because chemotherapy in and of itself is going to have more systemic side effects. But cancers can now be genotyped. So almost always you're going to get a pathology biopsy, a biopsy for pathology, a chunk of cells, and you're going to send them off for genotyping. And most times you're not going to do any treatment until they've been genotyped, because we want to know the gene mutations so we can directly, more specifically treat the cancer. And we give what we call targeted therapies. We can use things like the PCR, the polymerase chain reaction, and some other methods. Genetic abnormalities can affect patient outcomes, but can also really help determine therapeutic approach. The detection of the BCR-ABL fusion gene confirms the diagnosis of CML. So treatment with a specific tyrosine kinase inhibitor, such as imatinib, will switch off the growth-promoting activity of the BCR-ABL fusion protein. And because we now have this, we have seen a drastic improvement in the prognosis of patients with CML. I'd say before the year 2000 time, if you had CML, it was a pretty devastating prognosis. But now that we have the TKI drugs like imatinib, people are living and surviving CML. The other thing is these genes take time. The genotyping, takes time. And that can be very frustrating for the patient and the provider, because you're waiting 1 to 2 weeks, sometimes 3 weeks, to get these genotyping back. Since we did discover imatinib, we've now discovered it's helpful in other types of therapy as well. All right, HER2. HER2 is associated with breast cancer. It's a gene that can be active in some of the breast cancers. So you're either going to hear like HER2 positive or HER2 negative, because they want to specifically know if that tumor has that gene. If it does have it, if it's HER2 positive, we do have targeted therapies like trastuzumab and lapatinib to reduce the activity of HER2, and that can be a treatment to decrease the chances for metastases. We are going to touch on pediatric considerations because, as an acute care adult, quote unquote, acute care provider, we actually can go down to age 13. And so we do kind of dip our toes into adolescence. And so part of pediatric considerations still apply into adolescence. So it is important. And even though you are not going to be a true, quote unquote, pediatric provider, I do think this is pertinent to you and is only going to help you be a better provider. So we are going to talk about these things. The most common childhood cancers are leukemias and lymphomas, and the most common is the acute lymphoblastic leukemia, which is ALL. Okay. Most times, cancers in pediatrics are different from adults because it's not typically epithelial origin. So, oftentimes, the cancers cannot be explained by the environmental exposure. Okay. Most cancers are the hematopoietic, so lymphomas and leukemias. Then the next ones could be nervous system tumors or embryonic origin. Okay. Wilms tumor is a kidney tumor that has embryonic origin, and retinoblastoma is a cancer with genetic origin here. So really, when you think about childhood and cancer, leukemias and lymphomas are much more common. Most cancers in children are not from purely the environment. They haven't been alive long enough to really have their genes mutated from their environment yet. So these are typically more germline or just bad luck things, yes, but yep, it's there. Your book gives you signs and symptoms of cancer in children, and they call it the child cancer acronym, or mnemonic. Sorry, that's what I'm trying to say. And that's in box 7.2. But you have to think about it. They're not going to always present typically. They often have more of a bloodline type of cancer. So they have things like unexplained weight loss, headaches, early morning vomiting, increased swelling or persistent pain in their joints, backs, or legs, lumps or masses where they're not supposed to be, excessive bruising, bleeding, or rash. They are sick all the time. They can get a whitish color behind the pupil. They can just be nauseated and sick to their stomach, constant tiredness, notable paleness, vision changes, or fevers of unknown origin. Gerontological considerations are much more up our alley. So this makes sense now to say the human body, when you're a gerontology patient, you're old, right? So you have been exposed to your environment for a long time. So a lot of the cancers are favorable to environmental causes. A decline happens naturally with age in DNA repair mechanisms, and so that also puts us at a greater risk of having DNA mutations or bad genes that are more faulty and are more susceptible to mutation. There can be multiple sporadic DNA damaging exposures. We know that we've been exposed to viruses, free radicals, and carcinogens, and all of those can lead to the altered expression of oncogenes and altered expressions of our tumor suppressor genes, our apoptosis genes. We also know that we can have initiation of altered phenotypes with increased cell proliferation. We could accumulate additional gene alterations with age. With age, we tend to promote the transition to malignancy faster, with gradual acquisition of more invasive and metastatic capacity. And tumor expansion is supported by angiogenesis, all of which are occurring against the natural backdrop of a declining host. When factors favoring malignancy outweigh your factors inhibiting malignancy, cancer is what we get. And that's what this visual image is here. So there are some natural things that happen to our body with age, especially when we're talking about cellular level gene mutation, DNA repair. Those start to get, I would call, faulty the older that we get. So obviously we're also older, so we've been exposed to a lot more of environment. So those two factors also come into play with, we tend to be weaker, frailer, and more immunologic deficient, and not in as good of health with more disease states. So all of those factors together will definitely increase the chance of having cancer as you get older. I do want to talk briefly about the decline in the immune function, which is often called immunosenescence. So the immune system, obviously, we know, is a major defense mechanism against the development of cancer. And as we do age, we do have loss of immune function. Okay. We talked about this in the last chapters, that the thymus is the major site of T cell development and maturation. And we have that decline in the thymic output happens as part of aging. The decline in the functional immunity is more permissive to tumor formation, but also may promote it by contributing to chronic low levels of inflammation. Remember, we don't have as robust of an immune system, and we don't have as robust of an inflammatory response. And so we aren't able to trigger the release of these T cells and B cells and things to have our own immune system attack a cancer as well as it could when we were younger. We also have alterations in our hematopoietic stem cells. So the unique ability of our stem cells to proliferate, differentiate, and self-renew keeps us in a very nice level playing field, and we're pretty healthy because of that, right? We can replace cells that are destroyed because we have active bone marrow. It's a very coordinated process, and we keep this replication of our bone marrow cells very consistent. But with aging, it brings a cascade of changes to this process. We get a decline in organ function. That can affect our bone marrow and our hemopoietic system, especially our stem cells. So now, to replace blood cells that are constantly being lost due to destruction, such as through the spleen or tissue utilization, our hematopoietic stem cells continuously need to regenerate circulating cells of the blood and the immune system throughout our entire life. But our hemopoietic reserves are depleted during aging, and our ability to renew it deteriorates as well. So we lose the ability of the stem cells to differentiate into different cell types. There's a decline in stem cell functioning, but not necessarily in stem cell numbers. Because of the long lifespan and their ability to replicate, stem cells are subject to damage from both intracellular and extracellular sources. Okay? Intracellular sources include the oxidative chemical reactions that occur within the cell. Stem cells undergo repetitive DNA replications during their lifetime, and such repetitive replications can cause random errors. So over time, we've had more random errors than more number of years we've lived. So genetic damage can accumulate, and our instability of our stem cells exists to a greater degree in bone marrow of older adults. We know this, and that suggests we have a progressive decrease in DNA repair, and that leads to genomic instability with age. Damage to DNA occurs in the aging process because we have prolonged exposure to both internal and external agents. And remember, the DNA ability to repair itself is reduced with aging. So if DNA cannot be repaired, over time mutations accumulate, and that's going to increase the incidence of transforming these cells to malignant cells. This is often the process for why we get a condition in elderly people called myelodysplastic syndrome. And it really is a disease of older adults. And the easiest way to understand that is we just get failure of the bone marrow. All of these things we just said. We get this accumulated damage over time. Our DNA can't repair the same. Everything is just kind of faltering. And to me, it's like we're driving the car down the road and the rims are falling off, and we lost our bumper a while ago, and we've got a chip in the windshield, and we got scratches and a dented bumper, and a headlight is out. Like, we're still driving the car. The car is still working, but it's just beginning to break down. And that is very much what myelodysplastic syndrome is. We just get a failure of the bone marrow. And it's not necessarily a cancer. That's the other hard part. It could just be a process of aging. So it's not that we have created malignant cells, but we have damaged the ability to make our cells, and our bone marrow is so dysfunctional, it acts very similar to like a leukemia. And then with time, MDS, myelodysplastic syndrome, can over time then convert into a leukemia. This disease to have. Not everybody ages and gets myelodysplastic syndrome, okay? You're not guaranteed to have MDS by the time you're like 100. So it is still a pathological condition that can happen. My point is that it is a pathology. It's abnormal. It's not good, but it's not necessarily a true cancer in that it's not necessarily caused by a true neoplastic process, but it can quickly convert into one. The book also talks a little bit about some practice implications. To me, it sounds like, hey, we're saying the older you are, the higher the risk of cancer you have. Why don't we increase our surveillance for people? Because if you think about it, we have all sorts of things that you've probably heard where we actually age out when we recommend doing screening for things, right? So oftentimes, after a certain age, we don't screen for cervical cancer in women anymore. We reduce the frequency of colonoscopies. When you're talking about surveillance and some screening, we have a lot of data that drives that. That shows us that if you haven't gotten it by a certain age or what have you, then things are different. So it's not just because you're getting older, we should screen. We actually kind of have a lot of variables that are affecting that. Some of it is the functional status of the individual and what their prognosis is, their degree of frailty, and then also just their personal preference of what they're willing to go through. Older adults can be more frail, and they can have a reduced functional reserve, and so they don't recover well. They don't tolerate stress as well. So they aren't always going to tolerate treatments as well. They're not going to respond to the biggest dose of chemotherapy as well as someone who's younger. Older adults can have altered liver and renal drug clearance, and so we have to assess that, and that oftentimes adjusts dosages of our treatments. So, nonetheless, even though you're at higher risk for cancers as we age, we don't necessarily just start screening when age comes. The book also makes a good point of just saying it's all fine and good that we can come up with some of these novel cancer agents and we can come up with all of these great drugs, but we're still limited a lot of times because at the end of the day, the older adult just can't always tolerate things as well. And so that does also limit what we're able to do for treatment for the older adult. I like your book, as the chapters are beginning to progress, that they have something called Bridge to Clinical Practice. In fact, let me just add that here. Let's do aging and cancer. So you had a slide for the things I just talked about. But let's also talk a little bit about the Bridge to Clinical Practice. I'll just add that here. You have a spot for it in your notes. And I like this. So they start giving us history and physical examination, screening tests, diagnostic tools, some of the labs we could use, some of the treatment options. I want you to read through these, and please do. Please take the time to read them, but do not focus on them, okay? So the bridges to clinical practice are meant to kind of begin to give you the breadcrumbs, so to speak, of what's coming. As we move into your next year, this is essentially what we're doing, is we're building these, putting this all together. You are not at that point yet, so please do not read the Bridges to Clinical Practice and feel like I need to memorize everything that's happening, because that is not true at all. However, in years to come or semesters to come, you may want to be able to refer back to these sections in your chapter, and they may be very helpful. I like the way they're organized. Just don't get caught up on spending a lot of time on these for your studies. At the end of the chapter is also these key points again in your like little blue box. Read through these. If you want to just do a quick review of something and not go through all of your notes or whatever, just read the key points. They're really good, and they help very well, I think, in talking about the key points that this whole chapter has been. So this is the first section I was talking about. We've now covered the text of the book. The next part that I'm going to include in this module is now beginning to talk about some of the specific types of cancer that we may see in practice.